our body and the vertebrate body uses action potentials, which are a change in the membrane potential, as the way that we send signals through the neurons. And we talk about how the retina contains these specialized cells known as rods and cones, which are essentially neurons that send a signal via an action potential to another cell known as a bipolar cell. So if you remember the retina anatomy, right, is that the rods and cones are connected to the bipolar cells, and the bipolar cells then are ones that are going to communicate the signal into the optic nerve. So let's take a look at one of those. We're going to take a rod as our example, and because knowing the overall general form of the rod is very easy to convert over into the cones. Okay, so there is a rod, and inside of the rod there are going to be a couple of different things. The first thing that I'm going to put on here is I am going to put a sodium potassium ATPase, right? So this is going to move sodium out, and it's going to move potassium in. Okay, with the use of ATP, which converted into ADP and inorganic phosphate, right? So that's going to keep our concentration of sodium on the extracellular side high, just like we would in a traditional neuron. Now, the structure of the rods creates a folding of the cell membrane, like so. And on those folds are going to be some molecules known as rhodopsin. I'm just going to draw a couple of them, but this is sort of like studded with them. Okay, so this is what we call rhodopsin. Rhodopsin. All right, so the other thing that is going to be present on this cell is a sodium channel, but this sodium channel is a little bit different in that it is a ligand gated channel. Okay, but this ligand activated channel is activated by a molecule of, let's make that dark blue, cyclic GMP. Okay, so when cyclic GMP binds to this channel, sodium can flow through. Okay. The other thing that I want to put in here is a G protein. And this G protein is known as transducin. Okay? All right, now, here's where things get fun. All right, so we're going to bring in light, and what happens is light is going to take and hit this molecule of rhodopsin, and the energy from the photon is going to get a component of that rhodopsin known as retinol to change from its cis form to its trans form. So it's going to go from cis retinol to trans retinol. Okay? So when that happens, when that changes conformation, this molecule of transducin can now interact with that rhodopsin. And when transducin interacts with that rhodopsin, what happens is it produces and activates something known as cyclic GMP phosphodiesterase. Okay, so cyclic GMP phosphodiesterase. And as you might imagine, that cyclic GMP phosphodiesterase comes over and it's going to take that cyclic GMP and it's going to break it down. So if that cyclic GMP gets broken down, this channel becomes shut and sodium can no longer move through it. So our channel shuts. So if sodium can't move through that, what happens to the membrane potential now of this cell, right? So when this cyclic GMP gets broken down, sodium can no longer move through, the membrane potential goes down. That's a key component, right? So we've always talked about, well, action potentials are generated when the membrane potential goes up. But here we're saying when light is present, our membrane potential goes down. So let's take a minute and let's look at this interaction now between our rod and the bipolar cell. So we're going to move up a little bit. And what we're going to do is we're going to say, okay, we're going to draw our two cells now. We'll draw one like this. And then we'll just sort of draw the other one like this. Make it simple. Okay, so this is going to represent our bipolar cell. And this is going to represent our rod. Okay, now inside of this rod, there are vesicles. These vesicles contain a neurotransmitter. It's going to be blue. And that neurotransmitter is something called glutamate. Glutamate. Okay, now over here on my bipolar cell, there are glutamate receptors. Okay, so remember from our discussion on action potentials, a lot of times what happens when an action potential reaches the axon terminal, it's going to activate a calcium channel, voltage-gated calcium channel. Calcium is going to enter the cell, bind to that vesicle. That vesicle then will exocytose the neurotransmitter, and then the neurotransmitter can go over and bind to the receptor. Now, in this bipolar cell, the binding of this ligand to this receptor causes a signal transduction pathway which leads to the closure of a sodium channel over here. Okay, so binding of the ligand causes the closure of this sodium channel over here. All right, so now let's go back up here for a minute. Okay, stay with me. Right, when light comes in, binds to the rhodopsin, or hits the rhodopsin, causing cis-retinol to go to trans-retinol. Transducin interacts. We get the production and activation of cyclic GMP phosphodiesterase. Cyclic GMP phosphodiesterase breaks down the cyclic GMP, closes our sodium channel. Our membrane potential goes down. So if our membrane potential goes down, right, we are polarizing the cell, which means that this calcium can't bind to that vesicle because we're not activating this voltage-gated calcium channel anymore. So that's going to go away. Okay? So then what's going to happen is you're going to get no exocytosis of the ligand. The ligand cannot bind to that receptor. This signal transduction pathway does not happen, which means this gate does not get closed. And sodium can flow into the cell, and that's when we generate the action potential in the bipolar cell. Okay, so light coming in and hitting the rod, right, causes the inhibition of the action potential in the rod, but the rod elicits an inhibitory postsynaptic potential in the bipolar cell. So by inhibiting the rod, we do not release this inhibitory neurotransmitter, which means we stop the inhibitory postsynaptic potential in the bipolar cell. So by stopping the release of the neurotransmitter, we get the signal in our bipolar cell, which then can send the signal to the brain. So take a minute, right? I want you to draw out what sort of happens in the light cycle when light is present. Draw out what's happening, right? What's happening here inside of your rod, what's happening here at your synapse. Then do the same thing when light is not present, okay? Draw out what's happening in the rod, draw out what's happening in the synapse. And so when you get all done, your sort of image should look like this.